Hi, my name is Matilda and I've been doing a 10-week internship at Rothamsted Research. So I applied for this project because I've always wanted to know what it's like in the day-to-day life in the lab. And since I've always been fascinated in biology, I've wanted to know how to learn new lab techniques, and I found this project particularly fascinating. So the project which I was working on was on Zymoseptoria tritici, which is a fungal plant pathogen affecting wheat crops around the globe. And this causes a disease known as Septoria leaf blotch. And this results in necrotic brown blotches on the wheat's foliage. This is due to the fact that it secretes LysM domain effector proteins, 3-LysM, 1-LysM and X1-LysM, which essentially mop up or sequester any chitin shed by the pathogen. So since chitin is usually detected by the wheat, triggering defenses, those effectors enable Zymoseptoria to go undetected by the plant, and it also protects Zymo against any host chitinases. So this project focuses on whether proteins with sequence homology to ZT3-LysM would basically complement the function of ZT3-LysM and restore the virulence in any knockout strain. So the proteins were selected from a species with a wide range of lifestyles, and this is to generate complementation strains. And as this project is focusing on generating Gibson-assembled plasmids containing the ZT3-LysM promoter, the CDS or the coding sequence of each gene of interest, and then the ZT3-LysM terminator regions. And all the ZT3-LysM promoter and terminator regions are codon optimized to match all the coding sequences. And these will then be used for transformation into ZT3-LysM deletion strains. So to start off with, I conducted a lot of PCR reactions and gel electrophoresis tests just to test the primers and the coding sequences and see if they're effective. Then I bulk PCR'd them, gel extracted them, purified them, and just in preparation for Gibson assembly, where we basically attached all the different fragments together so that they can be aligned in a plasmid containing a PCGen backbone with kanamycin resistance. Then we inoculated them in E. coli, which took up the plasmid, and we placed them on kanamycin plates so we can tell whether or not the E. coli did take up the plasmid or not. And the colonies which have appeared to taken up the plasmid were chosen for mini-prep to isolate the clone plasmids from the E. coli, and then further checks in the form of restriction digests were performed later on in the project. So I have learned a lot in this internship. I'm very grateful to have this opportunity. I learned several new lab techniques which will help me in the future, and it helped me work independently in the lab. It also taught me what it's like to be a PhD student and a research scientist. So I'd especially like to thank everyone at Rothamsted and the BSPP for this opportunity.